Let's gonna start the last chapter of Mcat Bio General Review for the genetics chapter. So, fundamental genetics definitions. Alleles are different forms of genes. Dominant is only when one copy is needed to be expressed. Recessive is both copies needed to be both copies needed to be expressed. Genotype is a set of alleles one has, and phenotype is the physical expression of those alleles. So the physical observable characteristics, observable physical characteristics. Patterns of dominance. So these are the basic ones. So complete dominance is when one allele is dominant over the other. Big A, small a, you'll have the big A being shown. Codominance is when you have more than one dominant allele. So basically, it's like cows when you have brown and white spotting showing up, little patches. And incomplete dominance is there's no dominant allele. There's always going to be an intermediate if you're a heterozygote. The example is pink flowers. Penetrance is the proportion of the population which expresses the given phenotype, and expressivity is the varying phenotypic manifestations in the genotype. Different types of mutations. Silent mutations have no effect on the produced protein. You can have different you can have a substitution, but then if your protein isn't the central dogma is DNA to replication to translation to protein. If your protein isn't changed, then this is a silent mutation. No big deal. Missense is a substitution of amino acid into the polypeptide chain. Nonsense is a premature stop codon, which is placed in a polypeptide chain, basically non-functional protein. Insertion and deletion is resulting in a change of the reading frame. Duplication is a segment of DNA which is copied multiple times. Inversion is a DNA segment which is reversed. So taking, like, let's say 5' to 3', flipping it, and the 5' becomes a 3'. Insertion is when DNA is moved from one chromosome to another, and then translocation is actually a swap between two chromosomes DNA. All right, so now we have the different types of crosses. All right. So different types of crosses. Recombination frequency is the basically percentage which is corresponding to map distance. The closer two genes are, the more linked they are. So basically, the less chance they're going to be separated in prophase. You've got monohybrid crosses, which will be like, for example, homozygous dominant, capital A, homozygous recessive. Yeah, homozygous dominant crossed with a homozygous recessive. And, sorry, actually, this is two heterozygotes. So if two heterozygotes are crossed, you'll have a 3-to-1. Is that right? Oh, you know what? I'll go back and look at that. Monohybrid cross will result in a 3-to-1 phenotype, and a one. Yeah, this is okay. This is a heterozygote times a heterozygote. There's a 3-to-1 phenotype and a 1-to-2-to-1 genotype. 2 meaning the heterozygote genotype. Dihybrid cross will yield a 9-to-3 to 3-to-1 genotypic ratio. Very classical is Mendel's pea plants, color and shriveled peas. Evolution definition. So punctuated equilibrium is evolution that is usually thought to be slow but then rapid changes. So for a long time, a butterfly will look the same. All of a sudden, in span of basically a couple of years, the butterfly will change and go from like blue to red, and then it'll stay the same for a long time. Stabilizing is a narrow range of phenotypes. So basically, if you look at a graph, it's probably somewhere in the middle, 50%. Directional is when one's direction is selected. So the example that Kaplan uses is antibiotic resistance. If you put antibiotics on a petri dish, it's really the far phenotype that is having antibiotic resistance will be selected for, and everything else will basically die out. You have selection on one end. Disruptive is too extreme selected. So when Darwin was studying birds, he saw that seeds were usually small or really big, so that the beaks were small or really big. There wasn't no intermediate beak because there was no intermediate fruit, so the environment was selecting for the features. Divergent evolution is two species with a common ancestor that have become different. So you can look at like cats and seals. Both are mammals, had a common ancestor. Seals are living in an aquatic environment. Cats are living in a land environment. Parallel evolution is with two species with the same ancestry that evolved because of analogy. So you can maybe think of like, bat species. They're both bats, they're mammals, and they're evolving in the same area. And so they're functionality wise, they're evolving analogy because analogy is function. And convergent evolution is two species with no ancestral relationship become similar. So, like for example, fish and dolphins. They have no, fish are not mammals. Dolphins are mammals. However, both have fins and um, tails. This is an example of a cross. So AA times or heterozygous times homozygous recessive. You take one half will be A, and then one half will be AA. And you can go ahead and figure out what the ratios are.